This video will serve as a pre-lab discussion for exercise 3 on the usage of the hemocytometer. We have here the hemocytometer. So this is an example of a hemocytometer. The one that we will be using in the lab is of a different brand, but they look the same. So if you can observe here, the hemocytometer is actually similar to a glass slide, but a lot thicker. And then there are certain special depressions and grooves found at the center of the hemocytometer. So the hemocytometer is also named as such because it was once originally used to count the number of blood cells as viewed under the microscope. Like glass slides, you also need to use a cover slip in order to successfully use a hemocytometer under the microscope. So if you observe here in the center portion of this hemocytometer, you can see two small squares. These are actually the counting chambers. So each hemocytometer has two counting chambers. In this exercise, we are going to be counting the number of yeast cells as viewed under the microscope. So in order to be able to distinguish between living and non-living yeast cells, we need to use a dye. And this dye is called methylene blue. So first, we are going to be preparing a yeast suspension. That means yeast mixed with water. And we are going to be putting it using a micropipette inside a microcentrifuge tube. And we are going to mix that with 0.01% of the methylene blue dye. And the volume of these two should be in a 1-to-1 ratio. Take note, we are using a microcentrifuge tube with a maximum capacity of 1.5 ml. So we need to compute ahead of time what that 1:1 volume ratio will be in order for it to fit in a 1.5 ml container. And then after mixing the two yeast suspension and methylene blue mixtures, we are then going to use the vortex mixer to sort of homogenize and mix everything together. Once we're done homogenizing the mixture, we are then going to use a micropipette and aspirate 20 microliters from our prepared yeast and methylene blue mixture. And we are going to pipette that carefully into the groove found here on the hemocytometer. So making sure that the liquid that we are pipetting will not overflow or run onto the side grooves, as you can see here. So this is how the pipetted mixture of yeast and methylene blue would look like from the top. Here's how it would look like from the side. And then once we've done that successfully, we then slowly and in a level manner put on the cover slip. And it should cause the liquid in the middle to spread evenly. And if you notice here on top, the spreading should cover both of the counting chambers of the hemocytometer. There is a gap though between the middle portion of the hemocytometer and the cover slip, as you can see here, where the sample resides. And that gap is 0.1 millimeters in length or in width, meaning to say the distance between the hemocytometer's surface and the cover slip surface on top is 0.1 millimeters. So let us magnify one of these counting chambers. So it would look like this under the microscope. There are 9 big squares. Okay, you can see that it's sort of divided to 9 big squares. And the total dimension will be 3 millimeter by 3 millimeter. And these sort of bigger squares that I am highlighting here are the 4 bigger quadrants, which you may use when you count the number of cells, if the cells are relatively big in size. And then here in the middle portion, we also have several smaller quadrants. And these quadrants are used to count the cells if the cells are relatively small and only visible under high power. And you can see the dimension of the bigger quadrants here will be 1 by 1 millimeter, while the dimension of the smaller quadrant is 0.2 by 0.2 millimeter. So for the sake of reference, we are going to put numbers in these quadrants. So we're going to put it this way for the bigger quadrants and this way for the smaller quadrants. So we need to remember the positioning at least here. The same is true for the second counting chamber, the one on top. So it also looks like this. So yeast cells are very small, so I think for this exercise, we are going to be using the smaller quadrants highlighted here in green. Okay, so this is now a bigger view of that smaller quadrant. By the way, the large quadrant will also look like this, but a lot larger. So this one might be through the high power objective. So you would now see the yeast cells after you've stained them. And notice how some of them are colorless and how some are stained blue. The colorless yeast cells are the ones that are living or viable, while the ones that are stained blue are the ones that are dead or inviable. Notice also which ones to count. Okay? So when we count cells in the hemocytometer in the quadrant, we usually include in our count the cells that are at the border, top, and left portion of the quadrant, and exclude the cells that are on or beyond the bottom and right boundary of the quadrant. So if we count the number of live cells here, we have 1, 2, 3, 4, 5, 6, then 7, 8, and then 9, 10, 11, 12, and then 13, and then 14, 15, 16, 17 live cells in this quadrant alone. And for the dead cells, we have 1 and then 2 dead cells. And then you are going to need to fill the table, of course, after counting the cells, the viable and the inviable cells in all of the small quadrants, 4 per counting chamber. And there are 2 counting chambers in the hemocytometer. So you need to fill up this table and then get the average number of live and dead cells. And then once your data are in place, you may then compute for the dilution factor, which is just the volume, the final volume of your yeast suspension, okay, before adding or after adding the dye, that's the final volume. In our case, the yeast suspension plus the methylene blue, that's the final volume, divided by the initial volume of the yeast suspension, meaning to say just the yeast suspension alone. So that's how you compute for the dilution factor. And then to compute for the number of cells per milliliter, you need the average number of cells, living or non-living, times the dilution factor, and then times 10 to the power of 4 if you use the large quadrants. But in our case, we will be using the smaller quadrants, so do not use 10 to the power of 4. Instead, we will be using 2.5 times 10 to the power of 5. Now, it's very important to understand why we are using this number. And the key here is that 1 milliliter is equal to 1 cubic centimeter. I will not discuss this further in this video, but you have to find out first for yourselves why 2.5 times 10 to the power of 5. And then lastly, you will be computing for cell viability. That's the average number of live cells divided by the average number of total cells times 100. So this is in percentage form.